Every time scientists announce a breakthrough, a promising cancer treatment, a new vaccine, or a medication that could save millions of lives, there's often an invisible part of the story that most people never hear about. Before many of these discoveries ever reach human patients, they are carefully tested in laboratories. And more often than not, one particular animal plays a starring role: the humble rat. For decades, rats have helped scientists understand how diseases develop, how medicines work, and even how our brains learn and remember. Yet this raises an obvious question: why rats? Why not dogs, rabbits, monkeys, or some other animal? What makes these whiskered rodents so valuable to scientific research? The answer isn't as simple as because they're easy to keep. Today, we're exploring why rats became one of science's most trusted research partners, right here on History of Simple Things. At first glance, humans and rats seem to have very little in common. We walk on two legs, they scurry on four. We build cities, they sneak through them. But beneath the surface, our bodies operate in surprisingly similar ways. Rats have hearts, lungs, kidneys, livers, digestive systems, and immune systems that function much like ours. Their brains are organized similarly as well, allowing scientists to study memory, learning, addiction, anxiety, and even depression. Even more impressive is genetics. Rats share the vast majority of their genes with humans. While the exact percentages vary depending on how they're measured, enough of our DNA overlaps that many human diseases can also occur or be recreated in rats. This means researchers can observe how illnesses develop in a living body before trying treatments in people. Without these biological similarities, much of modern medicine would have developed far more slowly. One of the biggest advantages of rats is their incredibly short life cycle. A rat reaches adulthood in just a few months and can have several litters every year. Each litter often contains around 8 to 12 pups. This allows scientists to study several generations in only a few years. Imagine trying to understand how a genetic disease is passed from parents to children using humans. It could take decades before enough information is collected. With rats, researchers can observe multiple generations in a fraction of the time, making long-term studies far more practical. Another reason rats became laboratory favorites is that they're relatively easy to house. Compared to larger animals, rats require less space, eat smaller amounts of food, and adapt well to carefully controlled environments. Laboratories can precisely regulate temperature, lighting, diet, and other conditions. By keeping these variables consistent, scientists reduce outside influences that might affect their experiments. If every rat lives under nearly identical conditions, researchers can be much more confident that differences in results come from the treatment being tested rather than random environmental factors. Consistency is one of the foundations of reliable science. Many people think of rats as dirty pests, but scientists see something very different. Rats are remarkably intelligent animals. They can solve puzzles, remember complex routes, recognize individual humans, and even learn from watching other rats. Some studies have shown that rats display curiosity, cooperation, empathy, and social bonding. They've even been observed helping trapped companions instead of choosing immediate rewards like food. Because of these cognitive abilities, rats are ideal for studying how brains work. Researchers use specially designed mazes, memory tests, and learning challenges to understand conditions such as Alzheimer's disease, Parkinson's disease, autism, addiction, and traumatic brain injuries. Their behavior often provides valuable clues about how similar conditions affect humans. Not all laboratory rats are the same. Over many generations, scientists have developed specific strains that are genetically consistent. Think of them as carefully standardized populations. If researchers in Japan, Germany, Brazil, and the United States all use the same strain under similar conditions, their results become much easier to compare. Some strains naturally develop high blood pressure. Others are more likely to develop diabetes, obesity, arthritis, or certain types of cancer. Instead of waiting for these diseases to appear randomly, scientists can study them in predictable ways. Modern genetic engineering has made this even more powerful. Researchers can now add, remove, or modify individual genes to better understand what each one actually does. This is perhaps the most important question. Why can't researchers simply ask volunteers? The answer is ethics. Scientists cannot expose healthy people to unknown chemicals, dangerous viruses, or experimental drugs without strong evidence that they're reasonably safe. Before any new treatment reaches human clinical trials, researchers need to understand basic questions. Is it toxic? Does it damage organs? How is it processed by the body? What dosage appears effective? Animal studies help answer these early questions while reducing unnecessary risks to human volunteers. Only after passing multiple stages of testing can many treatments move into carefully monitored human trials. Laboratory animal research today is very different from what it was decades ago. Modern research follows strict ethical guidelines designed to protect animal welfare. Scientists follow what's known as the 3 Rs. The first is replacement, using alternatives such as computer simulations, artificial tissues, or cell cultures whenever possible. The second is reduction, meaning researchers use the smallest number of animals needed to obtain reliable results. The third is refinement, continually improving housing, handling, pain management, and experimental procedures to minimize discomfort. Rats didn't become laboratory animals because they were convenient pests that happened to be available. They earned their place because they offer an unusual combination of biological similarity to humans: rapid reproduction, intelligence, predictable genetics, and manageable care. Their contributions have helped scientists develop vaccines, antibiotics, surgical techniques, treatments for heart disease, diabetes, cancer, neurological disorders, and countless other medical advances that have improved and often saved human lives. At the same time, their role reminds us that scientific progress carries ethical responsibilities. Every experiment represents an effort to learn something important while minimizing harm, and modern research continues searching for alternatives that can reduce animal use even further. So the next time you hear about a breakthrough in medicine, there's a good chance that somewhere along the way, a small laboratory rat helped make that discovery possible. Sometimes, history's biggest advances begin with the smallest helpers. Thank you for joining us on this journey through the history of simple things. Don't forget to like, subscribe, and stay tuned for more stories woven through the smallest details.